We all know that DNA contains information regarding our genetic makeup and our phenotypic expressions. Using genetic engineering technique, it is possible to isolate and clone DNA. It is also possible to identify the sequence of the entire DNA present in an organism's body. Many genome projects help to reveal the genome sequences of different organisms. One such genome project is Human Genome Project, which we are going to discuss here. Human Genome Project was launched in 1990. It took 13 years to complete the entire sequencing of the human genome. It was a mega project involving many countries. The human genome is approximately 3 into 10 raised to 9 base pairs. The estimated cost of sequencing was $9 billion. High-speed computational devices were used to store the sequencing data. Why was the sequence data stored in computational forms? We know that nucleotide sequences are written in the form shown on the screen. If we have to store the data in conventional books, it would take 3,300 books, with each book containing 1,000 pages and each page containing 1,000 sequences or letters to store the information of DNA sequences from a single human cell. Hence, this enormous data was stored in computational form, and this gave the boost to a new field of biology called bioinformatics. The data stored in computational forms were easily retrievable, accessible, and analyzable. Let's now see the goals of Human Genome Project. The important goals of Human Genome Project were identifying all the genes in human DNA. A human DNA approximately has 20,000 to 25,000 genes. The next goal was to sequence 3 billion base pair that make up the human DNA without any error. The third goal was to store the information of sequences in computational database. The fourth goal was the plan to improve the tools for data analysis. The fifth goal was to transfer related technologies to other sectors such as industries. And the sixth goal was to address the ethical, legal, and social issues that may arise from the project. As I have mentioned earlier that it took 13 years to complete the project. The project was coordinated by US Department of Energy and the National Institute of Health. The Wellcome Trust became one of the major partners during the inception of the project. Later, many countries such as Japan, China, France, Germany, etc. joined the project. After the successful completion of human genome sequencing, many organisms such as bacteria, yeast, Drosophila, rice, etc. were sequenced. Let us now see what methodology was adopted to sequence the DNA. The methodology used for sequencing the DNA involved two major approaches. The first approach was express sequence tags, which refers to identifying all genes that are expressed as RNA. The second approach was sequence annotations. It is to sequence the whole set of genome with coding as well as non-coding sequences and later assigning the functions to the different region of the sequences. We'll now see the steps involved in sequencing the DNA. The very first step will be the isolation of DNA. Once the DNA is isolated, it is then converted into random fragments of small sizes. The fragments are then cloned in the host using specialized vectors. Can you guess why cloning of the fragments are done? Well, cloning helps to multiply the number of copies of the DNA fragments. Multiple copies help to ease the process of sequencing. The commonly used hosts for cloning were bacteria and yeast. Bacterial artificial chromosome, that is BAC, and yeast artificial chromosome, that is YAC, were the cloning vectors. Next is the sequencing of the cloned fragments. These fragments were sequenced using an automated DNA sequencer which was designed on the principle of the methods developed by Frederick Sanger. The sequenced DNA were then arranged based on the overlapping sequences in them. So, what do you think, was the sequences arranged manually or using computational programs? Manual arrangement of these sequences is not possible. Hence, highly advanced computer-based programs were used for this purpose. In the last step, the sequences were functionally annotated and assigned to each chromosome. The Human Genome Project got completed in the year 2003. But the completion of the sequencing of chromosome 1 was in May 2006. It was the last chromosome in 24 chromosomes to complete its sequencing. You might think that a human genome consists of 23 chromosomes, then why was 24 chromosomes sequenced, right? The answer to this is very simple. 22 chromosomes were autosomes common to every set of human genome. But the two chromosomes were the sex chromosomes X and Y. After the DNA sequencing, the next difficult task was assigning the genetic and physical maps on the genome. The maps were generated using information on polymorphism of restriction endonuclease recognition sites. Microsatellites, that is the repeating DNA sequences, were the second. We'll move on to the silent features of the Human Genome Project. The human genome contains 3,164.7 million bases of nucleotides. An average gene consists of 3,000 bases. The total number of genes is estimated to be much lower than the previous estimate of 80,000 to 1,40,000 genes. The functions are unknown for over 50% of the gene discovered. Less than 2% of the gene codes for proteins. Repeated sequences make up a very large portion of human genome. Repetitive sequences of DNA have no coding function, but they give the idea of chromosome structure, dynamics, and evolution. Chromosome 1 has the most number of genes, that is 2,968 genes, and chromosome Y has the least, that is 231 genes. Scientists have identified about 1.4 million locations where a single nucleotide polymorphism occurs in humans. These were the silent features of Human Genome Project. Let's now move on to the last part of this session and discuss the applications and future challenges of Human Genome Project. The knowledge about the DNA sequence will help us to get better adapted in the changing environmental conditions. All this knowledge can be used to face the challenges in various fields such as health, gene therapy, biotechnology, etc. The whole Human Genome Project took expertise and creativity of tens and thousands of scientists from various disciplines and from all over the world to achieve this task. It gave biological research an entirely different perspective. Human Genome Project helped in approaching questions systematically and on a larger scale. It also helped to study all the genes in a genome, the proteins formed by them and their interactions. We have studied the goal, scope and application of Human Genome Project in this session. Can you say how will the knowledge of human genome sequence help to identify the criminals or solve the biological parent disputes? Try to find out. Let's summarize what we have learned.
Manorama Horizon. Learning made simple.